Okay, maybe we'll get a start. So, welcome everybody to the this iteration of the Doherty Institute Seminar Series. It's my pleasure today to introduce Sefi Rosenbluh. He's from Monash. He's a Victorian mid-career research fellow there. He trained in Hebrew University of Jerusalem in Israel, and then moved across to Boston, where he worked at Harvard, MIT, and the Broad Institute. If any of you have had a bit of a look through his CV, there's a really impressive series of papers out of that time in Cell, Nature Genetics, really impactful papers in cancer. And then more recently, he moved back to Monash and joined the faculty there, where he's not only driving his own research lab, he's directing a functional genomics platform at Monash. And in particular, he's got expertise around CRISPR screens. And so he's been using that in terms of identifying genes that are involved in cancer risk. But clearly, the technology has a really wide application. So really looking forward to not simply seeing the biology, but also thinking more broadly about the potential of the technology that Sefi's developed and applies. I'm going to hand it over to him. I should note that feel free to put questions in the chat as we go through, and we'll try and get to them at the appropriate time, which may be at the end of the seminar, but if we see them at the right time, then we might be able to deal with them in real time. Without any more ado, I'll hand over to Sefi.
Thank you, Andrew. Let me just share my screen. Okay, so thank you for that introduction. And today I'm going to talk to you, and like Andrew said, I do have two hats. One is I run a lab, and the second I'm director of a platform that does functional genomics. And what I want to do today is take you through the technology that we use. Mostly it's CRISPR, but I'll basically genetic screens. And the theme of this, my lab uses these approaches towards questions in cancer, but of course these methodologies are not specific to cancer, and they could be used for basically any biological question. And what the take-home message I'll start with my take-home message from this whole talk is to show you the different types of technologies. Each one of them has advantages and disadvantages. There's no such thing as a perfect technology. And by knowing the problems and advantages of each one of them, it helps us to look at new questions, looks at new types of biology, and understand biology in a different way. And I hope that's what you take home from this. So the motivation behind everything we do in my lab, these kind of like genetic screens, is this. And this is a depiction of what we know about genes in the genome. And this kind of depiction of genes that we know something about is very underestimated, because in this representation, any gene that has one publication, regarding the quality of the publication, we consider that we know something about. And even if you take that very loose criteria, we still don't know anything about more than 40% of the genes in the genome. If you include in this non-coding RNAs, it becomes really sparse. And then of course, a lot of these genes are very context dependent, so it becomes even messier. So really the way to get and to understand how genes function is through genetic screens. Now, in the past, and I was involved with this in the past when we started doing this, what you had to do for a genetic screen is you had to create a library of reagents. So this could be RNAi or CRISPRs or any kind of reagents. So you had to create a whole array of libraries, so these plates, and you had to buy these very expensive liquid handlers. And even after you invested all that money and all that infrastructure and created all those reagents, every particular screen that you want to do is still extremely expensive, because you need the plasticware, you need a lot of people to do it, plating cells. It becomes a very big operation. But what I want to show you why the main technology that we use in my lab and in the platform is this fairly new technology of pooled genetic screens. It's come about in the past, I think, was it like 6 or 7 years. And it's really transformed this whole field of genetic screening. And here's the basic idea of everything that I will show you today. And the idea is that we could create this pool of lentiviruses. So this could be a pool of shRNAs or sgRNAs or ORFs. And the way we do this is we use the same technology that was previously used to create microarrays. We basically print, or not we have companies that do that print oligos on slides and then cleave them for the slides. And then what we could do is very quickly, with a few steps, we could create a pool of lentiviruses. So one tube that has tens of thousands or hundreds of thousands, depends on what we want to do, of lentiviruses, each with a different sgRNA or ORF. Now, the huge advantage of this is that it's very quick, very fast, and very cheap. So just in comparison, and I was at the Broad when we first made the first arrayed library of shRNA. So that was every well of a plate had a different shRNA. That was an effort that took more than a year to do. It had 10 people that were 100% of the time dedicated to that, cost millions of dollars to do, and was still not easy to screen with. Right now, with these technologies, we could create a pooled sgRNA library targeting all the genes in the genome with 100,000 genes. It takes us a few weeks to make and cost a couple of thousands of dollars. So that really allows us to modify this to different types of screens and different ways to screen. And this is how we typically screen with these. So we create this pool of lentiviruses, and then we infect a cell of interest at low MOI, so low multiplicity of infection. We ensure there's one sgRNA per cell. So here you have a collection of cells with one perturbation, and then we apply selection pressure. So this could be, for example, growth. This could be a drug. This could be gene expression if we're using FACS sorting to sort cells that have high expression or low expression of a gene. And the way we find our sgRNAs that are mediating the phenotype is by sequencing. What we do is we extract genomic DNA from the time 0 and the time after selection pressure. And we use this DNA to quantify, to count the number of guide RNAs. So in this example here, you see that applying the selection pressure caused overrepresentation of this brown guide RNA, right? And these other ones were depleted from the culture. So you could, the easiest thing to infer from this is, for example, proliferation. So in this case, you would say that guide RNA number, that had this pink guide RNA that targeted this cell, targeted a cell essential gene, and that's why it disappeared from this culture. So our first attempts with this was using Rnai. And here's the experiment that we've done in 216 cancer cell lines. So we've done genome-wide RNAi screen in 216 cancer cell lines. And what we could start asking is questions, for example, what are the genes that are important for a particular signaling pathway? And in this instance, we were looking at genes that are important in this context of Wnt activity. So Wnt, very well-known signaling pathway that is aberrant in many cancers and drives many cancers, particularly colon cancers. And what you could see here, if we take all, so the x-axis here is cell lines and the y-axis is genes. And what you are looking here is that genes that are important only in the context of Wnt activity. So when you have high Wnt activity, these genes are essential. So this is a scale of proliferation. Blue means that when you knock out this gene, the cells die. Red means that they don't die. And what we could find here when we differentiate these two populations is we find genes that are important in the context of Wnt activity. And of course, we find, many well-known, annotated genes that are important for the Wnt signaling pathway. But what was nice is that we could identify new genes that have not been previously connected to the Wnt signaling pathway, like YAP1, and we found this whole new pathway that is regulated in these types of cancer. So that was a nice proof of principle that this methodology could work. But what we didn't show in this paper is that one of the very not told and well-known secrets of RNAi is that there are a lot of off-target effects. That means we showed the genes that did actually validate, but there are a lot of genes in this list that did not validate, that even though we tried very hard, we could not ever validate their phenotype. And looking more into this, what we have found that these off-target effects are really, really driving these RNAi-based screen. So just 1-2 slides just to embark on this. What are RNAi off-target effects? So people use this term very a lot, but just to explain what they are and why they are important. So here's what you get when you finish an RNAi-based screen, right? So you get on the x-axis against cell lines. Each column here represents a different cell line, and on the y-axis you have different shRNAs. So all of these 4 shRNA would target gene A, or these 4 would target gene B. We call these shRNAs that are designed to target the same gene sisters. Now, what you would expect that sisters should be very, very similar. In this example here, you see that shRNA number 1 and 2 for gene A mediate kind of a similar phenotype. They have a similar pattern across the cell lines. And this is what we would want to happen. So if this was the case, this would be great. But when we actually look at the question and we ask how similar are sisters to each other, what we get is a very, very not overwhelming result. So this is what it looks like across, and this is now done across 500 cell lines. So here you're looking at the Pearson correlation between random shRNAs. If you take random shRNAs and you look at the phenotypic correlation between them, it centers around 0. That's what you would expect to see. They shouldn't be correlated. But then when you take these sisters, so shRNAs targeting the same gene, you get this orange line here. So you would expect to see, if these were really good, a very strong shift to the right. But that is not what we see. What we see is a very small shift to the right, almost pathetic shift to the right? Which this means that 2 shRNAs that were designed to target the same gene are not doing the same thing. And if you ask, what are they similar to? If you take 2 shRNAs, and here's a structure of an shRNA, we have these nucleotides within the shRNA between nucleotide 11 and 17, which we call a seed. Now, 2 shRNAs that were designed to target completely different gene, but all they share is these 7 nucleotides here. So they share the same seed. They, in fact, are very similar to each other. They drive a very similar phenotype. And what that tells us is that every time you're using an shRNA, you are doing 2 things in the cell. You are knocking down your gene of interest, but you are also inducing the seed effect. We don't exactly know what the seed effect does. The thought is that it regulates some kind of microRNA-based effect. But whatever it does, what we know, it's a very dominant effect of the shRNA. And every time you use an shRNA, you should be worried about this because you are doing 2 things in the cell. Now, there is a way to, a computational way to try and get rid of that effect. It's an algorithm developed by Bill's lab called Demeter. Demeter is the god of seeds. And the idea is that you use Demeter and it tries to manipulate and to remove that seed effect. But of course, every time you do such a harsh manipulation on data, it causes all sorts of problems and you lose a lot of information when you do this kind of harsh manipulations. So of course, we were very excited when CRISPR technology came out. Because that provides an alternative way to suppress genes. So the first CRISPR technology that I will introduce is what we call CRISPR Knockout. I'm sure everybody's familiar with. We use wild-type Cas9 together with a guide RNA. It binds to DNA, creates double-stranded DNA breaks, which will be repaired by the non-homologous end joining machinery. It'll create indels and stop codons and eventually knock out the gene of interest. So the first thing we did was to look at this technology. How does it compare when we look at cell lines? So here we are taking cancer cell lines that have mutations within these known pathways. The scale on the y-axis here is a proliferation. So meaning when you're looking at minus, means the cells are dying when you knocked out this particular gene. When you look at plus, that means that they are proliferating. And what you could right away see is that this is what we expect to see, that cells that have these cancer mutations are sensitive to sgRNA knocking out these genes. So that's very well. That shows us that we see an on-target effect. The sgRNAs are working. But what was more striking to us is when we looked at our negative controls. So these are, when we use, here we compared between shRNAs or CRISPRs. And we developed these 2 pooled libraries, each containing 150 either shRNAs, targeting, so these are targeting non-human genes. These are targeting luciferase, GFP, right? So these are genes that are not present in human cells. And what you would expect to see that when you knock out these genes in cells, nothing should happen to the cells. But what you see in these red dots here, when we look at these across these 10 cell lines, that a lot of these shRNAs have very dramatic effect on cell proliferation. So they're not inert, they're not doing anything. As opposed to CRISPRs, we were very surprised to see that we almost see no examples of these kind of off-target strange effect of these CRISPRs. So this was great, and this told us that CRISPRs have very strong on-target effect and a lot less off-target effect. But like I told you in the beginning, and I will tell this throughout my whole talk and all the time, also CRISPRs have issues. And here's the main issue that we have found with these CRISPR knockout screens. And this is a problem with cells, mostly cancer cells, but also not cancer cells, and this problem of genomic amplification. So here you're looking at an example of a pancreatic cancer cell line that has an amplification in chromosome 19. So here's the genomic landscape of this cell, and you see that in chromosome 19, you have this bump here, means that this region here in chromosome 19 is amplified. Now, when we look at a CRISPR screen across this chromosome, so each dot here represents a different gene on the chromosome, what you see is when we get to this region of amplification, suddenly it looks like everything here is essential for cell survival. So kind of there's a very strong correlation between this amplification and the dependency. So when you target a gene in an amplified region, it becomes essential for cell survival. In RNAi, you don't see that. There's no correlation between the dependency or the essentiality of that gene and its location on the genome. Another way to look at that is to just look at a simple correlation. The more copies of a gene you have, the more essential you are for that gene, right? So that obviously can't be something biological. That's an off-target effect. And when you think about how CRISPRs work, it actually makes sense. Because what happens is when you're looking at a non-amplified region, you basically have 2 copies of that piece of DNA. When you look at an amplified region, you have multiple copies. And now when you're inducing CRISPR, again, like I told you with RNAi, you're doing 2 things here simultaneously. You are knocking out your gene of interest, but you're also inducing a DNA damage response. And that will cause increased depth of these amplified regions versus these non-amplified regions. And that could be an issue, and especially when you're looking at proliferation as a phenotype. And actually, in some cancer cells, this could affect up to 30 to 40% of the genes in the genome, right? So this is probably the major issue we see with CRISPRs. To try and solve this problem, we looked at a different CRISPR technology called CRISPR-I. CRISPR-I, this uses the same Cas9. But here, instead of taking the wild type Cas9, we take a mutated version of Cas9. It's called dCas9. So this form of Cas9 has 2 mutations that abolish its catalytic activity. So it can no longer cut DNA. It can still bind to DNA, but no longer cut DNA. And the basic idea is that when we target this to promoters of genes, this will inhibit the transcriptional machinery. So you'll achieve the same gene knockout. So to test these 2 approaches, what we did is we compared between CRISPR-I and the CRISPR knockout that I showed you before. And here we used a different approach with this compound called 6TG. So 6TG is very often used in functional genomics. 6TG is a nucleotide analog, which in normal cells will become toxic. So 6TG is processed by a gene called HPRT1. And if you have wild type HPRT, the cells will die. But if you successfully knock out HPRT1, the cells will survive in the presence of 6TG, right? So what we are looking for is cells that survive means they have good knockout of HPRT1. And we created both CRISPR-I and CRISPR knockout sgRNA libraries tiling across the promoters and the exon of HPRT1. And when you look at it, here are the results. So here you see here, so what you see when looking at the bottom panel here, so this is the Cas9, and looking at the exonic regions of Cas9, so all the dots here, each dot represents a different sgRNA. And what you see when you look at the exonic regions is basically most of the sgRNAs, basically almost all of them, rescue the phenotype. So, and this is consistent with what we see with wild type Cas9, is basically it's very easy to find sgRNAs that work, because the majority of them just work. It almost doesn't matter where in the gene you target, they seem to knock out very well the gene. When we look at the CRISPR-I, we don't see that. When we target the exons, we don't see basically any effect of targeting the exons. Only when we target the transcriptional start site do we start seeing an effect with CRISPR-I. And the way we basically see this machinery, it's kind of like a fast driving train. So if you have a fast driving train and you want to stop it, if the train already started and you're standing on the railroads and trying to just stand there and stop the train that way, it's not going to work. In order to stop the train, you have to wait in the station and knock out the conductor. That's the only way you'll be able to stop the train. So this means that in CRISPR-I, in order to design guided RNAs for CRISPR-I, we have to identify and target the transcriptional start site. And now when we started doing this across many genes, this is what it looks like. So here we're targeting with CRISPR-I using 34 different cell essential genes. So these are genes that are essential for cell survival. We know that when we knock them down, cells should die. And so we see the same picture that I showed you before with the HPRT experiment, that when we target guides between -100 to 100 of the transcription start site, we find guides that show us the correct effect. But what you could right away appreciate that although we have guides that work here, we have a lot of guides that don't work here, right? So we're finding maybe about 40% of the guides that work. And that's just not good enough when we want to design guides for a screen, because if only 30–40% of our guides work, we won't be able to really get robust phenotypes. And what we hypothesized is that this is because we are not really correctly annotating the transcriptional start site. And in order to better characterize the transcriptional start site, we use this database called CageSeq. And CageSeq, developed by the Riken Institute, what the CageSeq does, it experimentally measures the transcriptional start site by capping the 5' mRNA and after protecting sequencing it. So you basically get a quantitative measure of the transcriptional start site. And even if you look at a gene, a very well annotated gene like MYC, this is what you will get. So here, if you look at the NCBI transcript of MYC, this is what it would look like. But if you look at CageSeq, there are basically 2 transcriptional start sites. The NCBI one is smaller. These would not affect the protein, but they will affect the 5' UTR. And what we find is that if we suppress transcriptional start site number 2, the lower one here, we see no effect, where you would expect to see a proliferation effect. When we target transcription start site number 1, the higher one, we see a very strong effect of CRISPR-I. And we could do this across, and we did this across 100 different transcriptional start sites, 100 different genes, and we find that always actually targeting the highest, the most robust transcriptional start site is the only one that gives good phenotypes. And I would say that NCBI is probably wrong in about almost 30–40% of their annotations of transcriptional start sites. So we find that CageSeq is a much more robust way to find the correct ones, which is very critical for these CRISPR-I experiments. So now that we know the rules for identifying CRISPR-I based guide RNAs, we can start using that for screening. And we've done that. And here is just to show you that CRISPR-I doesn't have the issue that I showed you before of DNA damage. So here you're looking at guide RNA targeting, in green here, negative control. So these are 1,000 negative control guide RNAs. So you can see that both the wild type Cas9, CRISPR knockout, or CRISPR-I, they center around 0, meaning they don't have any effect on proliferation. Core essential genes, so these are genes that are essential for every cell to survive. So these are guide RNAs targeting ribosomal, proteasomal genes, right? They have a very strong anti-proliferation effect in wild type, in Cas9 wild type cells. They do have a strong effect, not as strong with CRISPR-I. This is typically what we see, is that CRISPR-I gives a robust effect, but not as strong as CRISPR knockout. But what was interesting is that when you target this region AVS1, so this is a region that does not contain any genes. When you not target it, nothing should happen to cells. We don't see anything happen to cells with the CRISPR-I, but we do see some effect on proliferation in a significant effect with CRISPR knockout. So even when you just target, just create double-stranded DNA breaks, you already have some effect on proliferation. But again, like I told you before, and throughout my talk, every time, also CRISPR-I has issues. And this is the major issue that we find with CRISPR-I. And this is these kind of genes. About 10% of the genes in the genome contain this type of structure. and basically what you have one promoter that shoots in two directions. We call this a bidirectional promoter. And when you target CRISPR-I to this bidirectional promoter, it'll inhibit simultaneously these two genes. And this is an example of this. So when you see these two genes here, when you look at CRISPR knockouts, TRMT5 is essential for cell survival, and SLC38A6 is not. But when you look at CRISPR-I, it looks like both of them are essential for cell survival. And that is not because they both are, it's because the CRISPR-I, when you target this promoter, it simultaneously inhibits both of these genes. And our best conclusion is that if you have the ability, and I'll show you sometimes that we do that, to use both of these approaches, both CRISPR knockout and CRISPR-I, it provides really complementary information to each other, and it's a very good way to do these types of genetic screens. So now, by seeing this finding that CRISPR-I could work that way, it made us think, could we actually use that very narrow window that CRISPR-I has in order to look at some interesting biology? And here we collaborated with Patrick Tan from NUS Duke in Singapore, which he's interested in gastric cancer, and his lab did an experiment taking 17 different gastric cancer patients and using H3K4-ME3 nanochip-seq, looking at this histomark. So H3K4-ME3 is a histomark that marks active transcription. So when you would compare normal to tumors, you should see where, and compare them, you will find cancer-specific active transcriptions. This is what it looks like. You find basically three types of genes. First, most genes are unaltered. So if you look here at the gastric cancer or the normal cell, they're similar. You have one transcript or same transcript in both of them. But then you have some transcripts that have these somatic gains. So in this example here, you have an additional transcriptional start site. So in the normal, you have one transcript, but in the gastric cancer, you have two transcripts, or vice versa. Then in the normal, you have two transcripts, but in the gastric cancer, you have only one transcript. So this made us think that we can maybe use CRISPR-I to investigate which ones of these transcripts are important. So first of all, what we did is we looked at these transcripts, and what you could see is there are a lot more gained transcripts than lost transcripts. Only a small portion of transcripts are lost, but a lot of them are gained, of these gained transcripts. And what was interesting is when we use GSCA analysis, we find that these gained and lost transcripts are enriched with cancer-related, gastric cancer-related transcripts, and with transcripts that are important for developmental process or have these H3 these just transcriptional activation during development, which is an indication that these might not just be random events, but actually related to biology. So to look at that, if we could use CRISPR-I, we took this gene here, HNF4A. So HNF4A is a gene that has two transcriptional start sites. So this is what it looks like. You can see on the bottom panel here that it has P1 and P2, so two transcriptional start sites, which create two different transcripts. Both of them are expressed, so you could measure both of them. And then we use CRISPR-I targeting either the transcription start site number P1 or P2. And what we find is, as we've seen in our screen, our previous screens, is that when you target isoform P1, only isoform P1, and when you target this transcriptional start site, only this transcript is suppressed, and vice versa. When you target transcript P2, only transcript P2 is suppressed and not P1. So that tells us that we could use this methodology to look at these alternative transcripts and which one of them is important for these cancers. So we designed a pooled library that contains close to 9,000 different guide RNAs targeting either these altered transcripts or these unaltered transcripts. And we use these in 6 different gastric cancer cell lines to find which ones of these are important for cell proliferation. This is what it looks like. Just to show you the data, we find, as expected, most of the genes we find that are essential are pan-essential genes, but we find some transcripts that are important, these alternative, these gain transcripts that are important for, particularly for proliferation of gastric cancer cells. And here are the transcripts that after all our filtering, we found that are robustly active. So we find 7 different transcripts, which an alternative transcript is driving proliferation of a cancer. I'll just show you some of the data we have, validation of 2 of them. So CIT is a kinase. And here's CIT. It has two different transcripts. This transcript here, transcript P1, is gained in tumors, and we could validate that using PacBio, so long read isoforms. We could further validate the same phenotype that we saw in culture cells, also in mouse xenografts. So when we knock down either CIT with one of two sgRNAs and then implant these tumors into a mice, we see a dramatic reduction in growth. To look a bit at the mechanism of CIT, we knocked down CIT and used phosphoproteomics to look at proteins that are phosphorylated and important for CIT activity. And what we have found that a lot of the proteins that are phosphorylated by CRT are proteins that have been related to the cytoskeleton, the microtubule reassembly. Especially this gene here, and DRG1, which is a master regulator of the cytoskeleton. And you can see here that when we knock down CIT with these two sgRNAs, we see a dramatic reduction in NDRG1 phosphorylation. So that was nice that we could find a kinase that's important for cytoskeleton reorganization, and this alternative transcript is driving these phenotypes in these gastric cancer cells. But of course, the question is, could we target this? Because there's no available drug against CIT. And here we used a different platform that was developed at the Broad called the L1000 platform. There's a link here on the bottom. I very recommend everyone to look at that. It's a very useful database. And the L1000 database is basically a database of transcription signatures. So what the L1000 is a way to measure the transcriptional changes that are induced by targeting of, in this case, there's about 1 million different signatures there, different drugs. And what you could do is then take your gene of interest, measure the expression changes mediated by knocking out your gene of interest, and then putting that into the database and looking at drugs that are similar or dissimilar to that transcriptional signature. And when we did that, this is what we found, is that CIT signature, we found classes of drugs that are similar to CIT and classes of drugs that are dissimilar to CIT. As a sanity check, it was nice to see that tubulin inhibitors were very similar to the CIT signature, which is consistent with our findings that CIT is a regulator of the cytoskeleton. But what was kind of surprising to find that we find that PKC activators had a very similar signature. And we could validate these findings by a crystal violet assay, showing that when we take cells that are dependent on CIT, they're also dependent on these PKC activators, which suggests, of course, these are looking at very high concentrations, but it suggests a mechanism and something we're exploring. Could we find more genes in this pathway, in this PKC activation pathway, that are related to this CIT? A second type of genes that we find in these screen is an interesting class of genes. And these are genes in which we have two transcripts with opposing functions. The example of this is ZFHX3. So ZFHX3 is a known tumor suppressor gene. But to our surprise, in the genomic profiling in tumors, ZFHX3 is actually gained. So the opposite of what you would expect from a tumor suppressor gene, which you would expect it to be lost in cancer, this one is gained. But when you actually look at the transcripts, there's two transcripts, P1 and P2. The one that is gained is only transcript P2. This alternative transcript is gained. Transcript P1 remains the same. There's no change in transcript P1. Only transcript P2 is gained in the tumor. That's validated by PacBio sequencing. In our CRISPR screen, what we found is, again, that transcript P1 act like a tumor suppressor gene. That's what you would expect from ZFHX3, is that when you suppress it, you see cells grow faster. But when we knock down transcript P2, we get the opposite phenotype of cells now dying, not surviving. And we could validate that in lower throughput or in mouse models. And here's another way to show it with a colony formation assay. And when we knock down the tumor suppressor form, right, so transcript P1, we see colonies grow, so cells are growing faster, and we target the other transcript P2, the oncogenic transcript, we see less colonies, or hardly any colonies. And not only does this work on the colony formation level, but also on the expression level. So when we look at known targets of ZFHX3, so CDK N1A, which is a tumor suppressor, is as expected, transcript P1 upregulates it, and transcript P2 downregulates it, and MIB, an oncogene, the other way around. So this indicates that these two genes are working indifferent than each other. One of them is acting as a tumor suppressor, and the other one is kind of a feedback loop, which is suppressing it. This also correlates with patient survival when we see that patients with high levels of this transcript P2, so only this transcript P2, have a lower survival rate, which is nice to see. And we think that this represents a whole class of genes that work that way, in which one transcript performs a function, but a second transcript basically inhibits that same function. So kind of a way of the cell to self-regulate each other. And when that way is altered in cancer, for example, you get a cancer phenotype. So just to conclude this part of the talk, I showed you about alternative promoters, which are very pervasive in cancer. And now Patrick and his group developed new ways to look at alternative promoters throughout many types of cancers. And we see that this is a very pervasive and robust genomic feature of cancers. CRISPR-I is a way that we could inhibit these cancer-specific isoforms. And it starts now not only looking at genes, but now we can look at specific isoforms, which is really difficult to do with straightforward CRISPR knockout experiments. And I showed you two of these transcripts that are important. Now, I want to move on to a second project to show you how we are using CRISPR screens to identify breast cancer GWAS hits. So the main motivation for this type of screen is that the whole field of oncology now is moving to this personalized cancer medicine approach. With the idea is that we want to find genetic evidence or genetic abnormalities in a patient, and based on those genomic features of the patient, we want to develop a drug that targets this particular genomic alteration. And the reason that this is so important, not only for cancer, but for every disease, and why so much is invested in these GWAS, is that genes that we have genetic evidence that they are involved in the disease, drugs that targeting those genes are much more likely to become approved therapies. So it depends on the study you look, it could be between 3 to 8-fold increase in the approval of drugs that have genetic evidence for their involvement in the disease. And for that, our collaborators, so this is with together with George Yashenovich-Trench from QIMR, they have done genome-wide association studies in 100,000 different women that had breast cancer and 100,000 women that did not have breast cancer. And what they find is these variants. This is what these Manhattan plots look like. Of course, you find here a lot of genes that are very well known to be involved in breast cancer, BRCA1, BRCA2, ATM, and so on. But there's a lot of genes that we don't know nothing about. And in fact, most of them are actually not even genes. So if you actually look at the loci, at these variants that are associated with breast cancer, you could see that only a very small portion here in red, or the promoter 2, which we kind of know these are targeting actual genes, are in gene regions, but the majority of these breast cancer associated transcripts, and this is not true only for breast cancer, this is true for most US studies, are in intragenic regions. And we basically have very little idea of what these are doing. The thought is that these are regulating expression somehow, but what exactly they are doing, we don't know, because a lot of times the gene that is nearest to these variants is not the actual causal gene. So in order to look at that, Jono and George's lab developed an algorithm called Inquisit to try and predict these genes that are regulated by these SNPs. So here you see an example of how this works. So in red here, you see the SNPs, the cancer-associated SNPs for this region. And of course, there's many genes next to this region. And now we could use different types of readouts. So this is, we use ChIP-seq, chromatin interaction, or gene enhancer pairing. So different types of genomic connections. And based on this, we score each of these genes in this region. So each of these genes, based on these correlation, gets a score. And based on that, we say how likely this gene is to be involved in the phenotype. For the 196 loci, so each one of these collection of SNPs in a similar region is called a loci, we find 1,957 genes that are associated with these increased breast cancer risk. And the question is, what are these genes doing, and could we use CRISPR screens to actually find the genes that are connected to these loci? And the approach we took here was a systematic approach in which we took all of these genes and we created 3 different libraries. So I showed you before how we use CRISPR Knockout and CRISPR-I. So those are for suppression of genes. Here we took a third approach, which is CRISPR-A. So this is for activation of genes. So what we do is we create these libraries, we put them in these cells. So for all of these genes that are close vicinity to these cancer-associated loci, we are activating these genes and inhibiting these genes and measuring their phenotype. And here we're measuring proliferation in 2-dimensional cultures or 3-dimensional cultures in 6 different immortalized cell lines. And this is what this looks like. So here you're looking at the, this is one of these cell lines, an example. You're looking at the proliferation. So you can see that we find some very well-known tumor suppressor genes like TGF-beta-R2 or CREB-BP that are increasing proliferation. But we find a lot of really new and novel genes like NRIP1 or ataxin-7 or LPAR2, right, that when we knock them down are mediating proliferation. Similarly with CRISPR-A, so when we knock down with CRISPR-A, so here we're activating. We find less genes that when we activate, but MYC, very well-known oncogene, is found. Now, the advantage of using multiple different suppression approaches and multiple different assays is that we can start comparing between them. So although we do find a very strong correlation between 2-dimensional cultures and 3-dimensional cultures, so most of the genes do correlate, we do find genes that don't. And here's an example of one gene that we found, CFL1, that only scores in our 3-dimensional cultures, but not in 2-dimensional cultures. And we think that this is consistent with the function of CFL, which probably functions through modulation of the cytoskeleton network. And that's why it's only important for motility, and probably it's only important for 3-dimensional culture. That shows you, which always are with these screens, the more assays we use, the better we get at it. This is a summary of our proliferation screens. So what you can see is that we find a lot of new genes that are important. What is the nicest thing to see that Inquisit was very good at predicting. So about we saw a very strong enrichment in Inquisit genes in these cancer oncogenes. Means that Inquisit does a pretty good job in predicting which ones of these genes are regulated by these cancer-associated loci. Second phenotype we looked at, so this was proliferation in vitro. Now we're looking at proliferation in vivo. So here, the way we do this screen is we take our cells, we infect them with our sgRNAs, and after we let them proliferate a little bit in vitro, we implant them in immune-deficient mice. We wait for the tumors to grow out, extract the tumors, sequence, and that's how we look at the guide RNAs that are there. What we found, obviously, we find a lot less genes. Most of the genes that we found in our in vivo screens also score in the in vitro screen. But interestingly, one of the genes, DUS4, only scored in mice, but not in the 2-dimensional cultures. And what we thought that might not only due to the 2D versus to the in vivo versus in vitro, but it might be due to the fact that in vivo screens, we had to introduce MECDD into these cells, so to activate another gene in these cells in order for these screens to work. And that's exactly what we found, is that when we take cells that don't have MECDD, knockdown of DUS4 does nothing, but when we have cells that have activation of MECDD, DUS4 causes this effect on proliferation. And we could confirm this by looking at MECDD. So cells that have activation of MECDD also have activation of DUSP4. And what tells us that this is this feedback loop, and this already is important because this gives you now a potential way to treat these patients. So what this says is that women that have this particular variant that causes downregulation of DUSP4 will only have a cancer in the presence of activation of the MAP kinase pathway, and potentially we could intervene by using MAP kinase inhibitor to inhibit these cancers driven by this DUSP4 MECDD phenotype. A fourth phenotype we looked at is DNA damage. So here we use an olaparib synthetic lethal screen. And what we do here is we take the cells, and after infection with our library, we treat with olaparib with DMSO. And what will happen is the olaparib induces DNA damage only in the presence, will induce cell death only in the presence when you knock down a gene that's important for the DNA damage response. This is what we see here. Just to go over this quickly, we find a lot of very well-known genes in the DNA damage pathway. We basically can almost rediscover the DNA damage pathway, but we find a lot of new genes that are interesting. Here's a validation of some of these new genes that we found that are important for the DNA damage pathway. And again, this suggests a therapy because that means that women that have these alterations in these variants will downregulate these genes, and potentially you could treat these women with a PARP inhibitor, which are becoming very common in breast cancer. So now just the last part that I want to show you here is a new type of CRISPR screen. And everything I've showed you now is we're using this CRISPR screens to look at genes and the phenotypes that the genes mediate. But what we also want to try and do is connect between these candidate enhancers in a gene target. So we showed phenotypes. We could see we have an algorithm that predicts that they connect to each other, but could we actually use CRISPRs to look at a physical connection between these enhancers or these non-coding genes and a gene target? And here we use a method called CRISPR QTL. And the basic idea of this method is that we infect cells, and here we do this at high MOI. Right? Because, and the reason you do this high MOI is just because of cost, because we're using single cell, which is very expensive. So we want to maximize our ability to do this. And we infect cells with up to 10 sgRNAs per cell, and using a very particular vector. And then what we use is single cell sequencing. So we get the identity of the guide RNAs because they are they have a poly A tail, and we get the whole transcriptome. So we can basically ask, when we target this enhancer, what are the genes that are inhibited? Because we think that this gene will connect to the enhancer. And first thing we did is to look at, as a sanity tech, at guide RNAs targeting transcriptional start sites of known gene, and that works. We could inhibit these target genes, so this method works and is very robust. But when we look at enhancers, this is really nice to see, and I'm just showing you one enhancer here. So this enhancer here on chromosome 12 is one that all these SNPs here showed a correlation with breast cancer risk, but we don't know which SNP is important. And there's basically two regions here in this region. Our phenotypic screen found ATF7IP as one that's driving very strong phenotypes of proliferation, both in vitro and in vivo. And when we look at this CRISPR QTL, what you could look at is that guide RNA is targeting region 1 or region 2. So when we look at all these other genes in this region here, none of them seem to have an effect. When we target either region 1 or region 2, nothing happens to the expression of these genes. Well, ATF7IP is only inhibited by guide RNAs targeting region 1, the only one in this region. And we can validate that using high chip. So we see a connection between region 1 and ATF7IP, or by luciferase assay, which we find 2 of the variants in this region, which are driving the phenotype. So what I've showed you here is that we could use large-scale screens to identify new genes and phenotypes that are genes. So that's important because we can start looking at mechanisms and how these genes are working. And I've showed you that we could connect now between a variant and a gene, which allows us to take this whole GWAS pentagram to the next level of finding drugs that inhibit these GWAS hits. So with that, I would like to end. And again, just say that the main message of this talk is that none of these approaches are perfect. Each one of them has issues. And it's something to always take into consideration is that the more phenotypes we look at, the more readouts we have, and the more assays we use, we get a lot more robust phenotypes. And I would like to thank people in my lab and other labs that I discussed here that are involved with the screen, and of course, my funding agency. And I will be happy to take any questions.
Cool. Great, Sefi. Just a fantastic talk. Really, really interesting work. I can't see questions here. Should I be able to see questions somewhere? Not sure if people have questions. How do I see this? Chat, questions? Anybody got any questions? I'm sure this part of it is working. So no questions have been submitted. Okay. So should people put their hands up for questions? Is that, I would think, just raise your hands. I think is probably the best in the chat function. This is probably the best way to do that. So if you like, the data are really interesting, the pros and cons of each of those screens. If you were starting from scratch for a sort of a project, what would you do now, I guess is one of the questions. Would you start at the end or start at the beginning?
So I think we find that Crispr Knockout gives the most robust phenotype. So if we have one to do, we typically do that for only doing one. But in some cases, I think CRISPR-I has a lot of advantages. But if you want to do one, I would do the Crispr Knockout because we find the most robust phenotypes with it.
Now, the other question I was thinking about is, like, in a lot of your settings, you're looking at cancer outgrowth. I guess we're a place that's really interested in the immune system and the immune function, and clearly, in some cases, the immune environment is really a mixture of two sets or multiple sets of highly variable genes, and so being able to target one is not necessarily actually the answer. It's the matrix effect of both of those genes. Are there technologies coming online that allow you to, I guess,
target subpopulations?
Work out how, like interacting, like it's polygenic effects. How do you uncouple polygenic effects in this sort of system? So yours is still biased towards single gene outcomes. That's what I understand.
So we have actually systems to target polygenic effects. So we could target multiple genes. It is, to do it genome-wise is basically impossible. But we could do, we have now approaches to do, I didn't show this here, but we could do combinations of up to about 300 genes, something like that, right? Combinations like that. So you're doing all pairwise combinations. More than that, it becomes too big.
Yeah.
But it's getting there. Some of the technologies that are with, I showed this here with the Cas12 are very promising with that.
Cool. Damien Purcell's asked, are there therapeutic applications using the CRISPR approach rather than trying to find a small molecule drug? Could these be an application for future manufactured RNA?
I think there are. So most of the applications that are now being used are with CAR T cells. A lot of people are trying to use CRISPR-based approach, or are using CRISPR-based approaches. With the Cas9s, it's the problem with the Cas9 that it's really the one that we use, the S pyogenes Cas9, is that they're really large. So to use it in therapy would be like directly would be pretty difficult. But there are other versions of Cas9, like the Cas12 and a few other ones that there are, I think, a couple of companies that are trying to use them directly to target genes.
Cool. Not seeing any other questions floating around. Any hands? No hands? I guess the really, I'll do the ad. So if we wanted to access these screens, but I assume we could talk to you at some stage or engage with you in terms of
Yeah.
access to genes. I think some of the genes that are really interesting in the cancer space are probably quite interesting in context of viral infection.
Oh, definitely. And we obviously, we do in the platform, especially, we do these screens in many, I mean, my lab's interested in cancer, but we do use the same approaches for viral infection. Viral infections actually is very good because you could look at rescue of phenotypes, which is a very good phenotype to look at. So yeah, definitely.
Okay. Well, looks to me like we're just about done. So again, I really want to thank Sefi. Like it's a really, really impressive and thorough use of, you know, cutting edge technology, and we can see the sort of the benefits of using these parallel approaches. So I would encourage you, if you actually probably go home and think about, you know, what can this do in our systems? I think it's definitely worth some prolonged thought and some consideration of having some conversations with people over the river.
I definitely think so. I'm always happy to talk.
Thank you very much.
Thank you.
Thanks everyone. Thanks for rocking up.